So we're going to jump into our next lecture. This is the ecology and cyanobacteria toxins, and Anne will be leading this one up. So take it away, Anne.
All right. So, you know, this stuff is complicated, right? But the whole point of the whole thing is to understand how these things are interacting in the environment and how to manage them, right? How to effectively manage them or not manage them, depending on what your decision is. And so this primary part of my lecture is essentially what are those things that contribute to what you actually see in the lake, and then a quick review. And this is a real this is definitely a satellite view of cyanobacteria toxins, what the basic categories are, what their effects are. So the really important thing to understand is a lot of times with population dynamics, it depends on where you are, okay? So we all have to use the same wording. So phytoplankton are algae that are suspended up in the water column, and periphyton are attached algae. And then there's this in-between group called tychoplankton. And tychoplankton is essentially bottom algae that gets moved up into the water column when there's a lot of turbulence, or there's been an event like a flood or a geologic event. So phytoplankton or suspended algae, you can see blue-green blooms, cyanophyte blooms in large rivers. It happens a lot in Minnesota, actually. You can see them in slow-moving streams. Periphyton are important not only just in streams, but also in the littoral area of lakes. And we don't do a good job with periphyton. We don't do a good job with littoral algae in general. So also important are stratification schemes. So if, and that's affected by a lot of stuff. So first thing is, okay, if you're sitting where I am, I'm sitting in southwestern Michigan, we generally have our lakes set up in the spring. They're stratified over summer period and they fall out again in fall. So it's isothermic from the winter into the spring, and then the lake starts to set up. That's based on how deep you are, how much wind action there is, what your sunlight regime is like, where you are in the country or the world, what your water chemistry is like, and what else is going on in the watershed. Because if there's drawdowns or there's other things happening, then that's going to affect how strongly you can stratify. If we think about lakes that are in, typical what we think of a diamantic lake, you have an epimnion set up in the fall or set up in the spring, stays there all summer. This is where the bulk of the action's happening. You might get a deep water metalimnetic pulse, and then down here, if it's deep enough and there's enough going on, your hope is that it doesn't go anoxic, but often in deep lakes anyway, down towards the bottom it's anoxic, and very productive lakes at the bottom it's anoxic. So the chemistry at this bottom part is very different than the chemistry up here, and therefore the pressures on the algae are going to be very different as well. So the processes that affect population dynamics, kind of like yin and yang, right? There's always, what you see is always the result of a growth and a death, always a growth and a loss at the same time. So I think one of the most interesting things when I do outreach in 6th grade and I talk to kids, well, when do plants photosynthesize? They had no problem going during that daylight when there's light out. And I said, so when do they metabolize? And they always raise their hands and say, when it's dark out. And well, not so, right? You're always metabolizing, and then you have growth processes going on during a certain part of the day. So those two things are always happening, growth and loss at the same time. So when we're talking, and I think I need to do that. Sorry. So for growth processes, primary production, right? You're controlled by light, temperature, nutrients, algal physiology, how healthy you are, and competition, right, between different taxa. Heterotrophy, so that means you're augmenting primary production, and that's dependent also on physiology and environmental conditions. It's often cued by environmental conditions. Release from sediment, so that's going to be not so much of a growth thing as a importing and augmenting the population. From resting stages, could be turbulence, different life stages, and colonization, which is coming from upstream or an external source like ship ballast water or proverbial duck feet. So let's talk about each of these just a little bit. So primary production is adding biomass. It often, you're going to have the highest productivity at intermediate light levels. That's, basic pattern throughout biology, right? Often you have the most of something at an intermediate level, not necessarily super high and not super low. It is impacted by nutrient availability, by physiology. So if you are not very healthy, you are not going to be accumulating carbon at the same rate as if you were super healthy, right? Or if you're in the beginning part of your growth phase versus the end of your growth phase where you're close to senescing, your primary productivity rate is going to be different. Heterotrophy, I find this the most fascinating part about algal ecology, and that you think of heterotrophy as, okay, phagotrophy, right? I am a cell and I'm going to eat another cell. So in the case of denobrium, it's a chrysophyte. You cannot grow denobrium azenically. It has to be bacteria in the water column with it, and they phagocytize bacterial cells. Serratium, believe it or not, as well as other thecate dinoflagellates also are phagotrophic. Same with Cryptomonas, it's phagotrophic, has a gullet, very protozoan-like, and it can engulf picoplankton. It can also absorb nutrients directly from the water column. Neetsia is a diatom, both paraphytic and planktonic diatom. It can absorb organic compounds from the water column. So it doesn't necessarily just have to be eating something else. They can actually absorb organic compounds, whereas most plants need inorganic compounds, right? They need inorganic NO3, they need inorganic P. These algae can either take advantage of phagocytizing other cells or absorbing organic compounds. This just is super cool. So and I don't have any good photographs or images of this. I've been looking for a video for forever of thecate dinoflagellates engulfing other cells. Serratium can do this with Fragillaria crotonensis. So they essentially exude this organelle called a pallium. It engulfs often chain diatoms. They suck out the contents and jettison the pallium. Pretty cool. And this is a common strategy that's used by some of the thecate dinoflagellates. Releasing from the sediments can actually be pretty important. So we think of it most often with cyanophytes, cyanobacteria. So we have aconites, which are the resting cells of the Nostoclean cyanobacteria. But there's also cysts for things like Serratium, Hormigonia for the Ossoteralis cyanobacteria. So Hormigonia are very short sections, 6 to 8, sometimes up to 10 cells, very dense, and they'll go down to the bottom. The rest of the the sheath is gone, or there's a short bit of sheath, and they'll hang there until conditions are good, and then they'll start to grow, or in the case of aconites, they'll germinate. Also, chlorophytes and chrysophytes also produce cysts in oocysts that persist in sediments. I would say I've looked at core samples. I do aconite analysis in core samples. Aconites survive the best. I do see occasional dinoflagellate cysts, not very often. The other thing I see that are not live are chrysophyte cysts, like Malamonas cysts and cyanophyte cysts. I rarely see them live beyond a couple decades, 3 decades, whereas I have seen what appear to be fully fluorescing viable aconites from Dolichospermum and Vanizominin, hundreds of years old. It's very cool. And then or they get entrained from this flocculent sediment when there's turbulence or turnover. So they're either growing and then progressing up through the water column because they're getting buoyant, or they're getting washed up into the water column. Or your ability to maintain position in the water column. So this is Botryococcus up here. It produces its storage products as oils, and it uses those oils to be more buoyant in the water column. At one point, they tried to use them for biofuels. They're not very amenable for biofuels. Takes a few too many steps, but there's a lot of oil there, and that helps them stay buoyant in the water column. Something like Gliotrichia, see this refractive part, those are all the gas vesicles that are in the terminal part of the filaments. Not only that, but by having this kind of round conformation, it makes it harder surface area wise for it to sink. So you can produce oil, you can get an advantageous length to width ratio, you can produce gas vesicles and aerotopes. So there's, or you can be modal, right? Colonization can either be natural, water spray, aerosols, floods, stream flow from upstream, waterfowl, duck feet, right? They actually have a lot of water that's entrained up in those little feathers, and they can carry things from system to system. Amazingly, there's a lot of stuff in spray and water aerosols up in the air. I'm always amazed when you do long-term collecting, almost like a spat sampler in the water. There's quite a bit of algal cells that are hanging up in the water column that get moved around during large storms, or it's anthropogenic, right? Ship ballast water, that's the one we all think about. Canals, Welland Canal, should I say more, or something like live wells and engine manifolds. So jet skis, they encase water in the manifold that doesn't empty when they take the jet ski out of the water. They move that from system to system. It's easier to clean the hull of a boat than it is to clean the manifold of a jet ski. So those are big issues. And then of course, folks who will dump their live wells when they're done fishing for the day. When we talk about loss processes, so things like, sometimes you just die, physiological mortality, it's inevitable, and it's highly variable. So often, either it's cued by change in environmental circumstances, it gets cold, it gets cloudy, there's a lot of rain, the stratification starts to break down and you get moved out of the light, or it's something like part of the population starts to senesce for whatever reason, and there's actually chemical cues they share, and then everybody senesces at the same time, almost like a lemming going over the cliff, right? One goes, they all go. Grazing, really complex interactions with grazing. Sedimentation or burial, so hydraulic washout and scouring. This is especially important in man-made systems and reservoirs. And desiccation. Turns out you can actually use desiccation to preserve things for long-term archival purposes in herbariums. So you can tell when when a population is beginning to senesce. In the case of dinoflagellates, you'll start seeing cysts, empty theca hanging around. Denobrian, you'll see that some of the loricas have monads and many don't. So if you start to see this happening, you know the population's getting ready to senesce for the season. It's not like there wasn't something here and then all of a sudden it will get populated by a monad or by a single cell. The cell actually develops with the monad or with the lorica around it. So it's not like there's a new cell that's going to come and populate that empty lorica. Once it's empty, it's empty. So these are clearly senescing colonies here. Pediastrum and all of the new genera that go with that. Myoctinus and Pseudopediastrum, you can start to tell when you start to see empty cells here that the colony is starting to senesce, because it's not like a new cell comes and occupies that empty cell wall structure. It all develops together. Grazing, one of my favorite far sites. Broke my heart when he retired. So it's interesting because grazing can be it affects both quality and quantity of algae, because of the effect of grazers have preferences and they avoid or seek out certain foods. So grazing would be considered top down, whereas you have primary productivity with the nutrients and light and temperature, that's bottom up. Consumption and nutrient regeneration can both be a factor, and it's pretty interesting how this all plays out. So food webs are complicated, right? Whenever you're talking about who is eating who, in order to make this so that we can actually understand what this says, all of our nice little arrows go one way, but in fact, these arrows go multiple ways all over the place, right? Even fish will eat young of the year to some extent. So top down versus bottom up are like growth and death happening all the time. Depends on how big you are. Size does matter. How selective you are. Calanoid copepods are going to be less selective than a cyclopoid copepod. How dense you are. So very dense grazers are going to have more impact than non-dense grazers. What your specific excretion rates are. So think about, filter feeding is a specialization of grazing, right? And so zebra mussels will, and I'll talk about this in just a second, but they excrete at a very specific ratio, and essentially how you graze. So are you generalists like a Daphnia, or in terms of size, rotifers, their corona will just bring whatever can get into their gullet, whereas a cyclopoid copepod that is a little bit more selective about what he goes after. I always, because I have 4 kids, so I always think of cyclopoids as being teenage boys, and the calanoids, or the cyclopoids as being teenage girls, and the calanoids as being teenage boys. Calanoids will go for whatever they can get. Key algae features, so what your growth rate is. Do you have resistance to grazing? Do you have resistance either because you're big or because you have structures that make it hard? Is there allopathy going on? So the big question about algal toxins, right? Is there a reason for that? And is that supposed to deter grazing? And your ability to migrate. You know what? If just like snowbirds, if you don't like the current circumstances, leave. So migration is a way of leaving, right? So if you're growing very fast, that's going to help overcome grazing. If you are big, it's going to keep most of you from being grazed, like large Phanosomnium fascicles. These are grazed Goliatrichia colonies, that haircut that Ken refers to. This is daphnid, actually, grazing activity. For daphnids, if they're too complicated, like a Phanosomnium, like big Dolichospermum colonies, it clogs their gullet, and they have to try to use their post-abdominal claw to clear that. That causes problems for them. They are not effective grazers in the presence of Microcystis, Phanosomnium, and things like that. But they'll eat other things, and they'll eat as much of the other things as they can get into their gullets. If you have big spines, something like Rhizolenia or Acanthoceras, Urosylenia have big long spines, makes it pretty hard to get you in their gullet. Something like Chrysosphaerella is a chrysophyte, colonial chrysophyte. Each cell has 2 waving spines. You get to a certain size, those just aren't edible. You're not going to be able to get them in your gullet. But generally, it's considered that anything under 30 microns is edible. But I've seen rotifers spin up Closteriopsis or Monoraphidium, almost like spaghetti, and get it into their gullet that way. That far exceeded that 30 micron limit. It turns out, just like our food, algae have different nutritional values. So the highest nutritional value is actually considered to be a cryptomonad. Cryptomonads are there all the time, but they're often kind of low level, except for very certain pulses in the spring, sometimes in the fall, occasionally over winter, but they're kind of always there. Make really good Daphnia food. Diatoms, because of the oils, are good food as long as they can get them in their gullet. Green algae are the next best, although often they can pass through the gullet completely. And blue greens or cyanophytes are the least nutritional quality. So, and then in response to that, algae can have no response, which means they are not a target. They aren't grazed, and they don't benefit from other things being grazed. They increase. Something like Sphericystis that actually passes through the gut undeterred, yuck, and makes it out better on the back end, so to say. Decreased, you are totally susceptible to grazing. And then a unimodal response, which means down here, there's other food the zooplankton prefer. And so they leave you alone until they use all that food, think deer and woody browse. So they use all that food and then they switch over to you, and then you have a negative response to grazing. So it's complicated. Microcystis has been associated with blooms in Lake Erie with the onset of the invasion by zebra mussels and then quagga mussels about 15, 20 years later. So it turns out zebra and quagga mussels don't like microcystis, so they actively reject it, and they eat everything else. They eat all the other algae that are present so that they take all the competitors for microcystis away, and they excrete at a nutrient ratio that's perfect for microcystis. So pretty commonly when you see big populations of zebra quagga mussels, you might have a short-term few year clearing phase, and then all of a sudden you'll start to see horrific microcystis blooms. Is not just happening in Lake Erie, it's happening in other lakes as well. Sedimentation burial has a lot to do with stratification, turbulence, sediment load. If you have a reservoir, you've got a much higher sediment load than an oligotrophic system. Less healthy cells sink faster. So if you start to see a lot of epiphytes like choanoflagellates and chytrids on diatoms, that tells you, okay, they're infected, they're going to go down pretty fast. How modal they are, they're less likely to be buried if they have either a rafe, which allows them to climb back up to the light, or they have flagella like chrysophytes, cryptophytes, dinoflagellates, things like that. They're going to be able to migrate up and keep up in the water column to some extent. Sedimentation will not be as important for those taxa. Hydraulic washout and scour, so that's very much an effect in large rivers and reservoirs, and that has totally to do with the flow, rainfall morphology, and the velocity of the water. And again, whether these mats the flow is high enough and they're at such a growth place in their growth phase that they will detach from the bottom and get washed up in the top. It's not unusual actually to see things like Didymosphenia whole, like all the sheath come downstream in a big flooding event, and just how old the mat will be. Desiccation, so you know, it acts as a loss process technically, but as Ken was saying, the cyanobacteria figured out, well, you know, just dry me out, I'll hang maybe for a few decades or a few hundred years, and then, you know, things get better again, and add some water and voila, I'm going to regenerate and grow just as well as I did before. It's pretty amazing, actually. So it has a lot. It's a great way to, if you can survive being taken out of the water column by one of these activities like an earthquake or a volcano, you know, subsidence of flood waters, and then, you know, wait for it to get wet again and come back again, ephemeral streams and rivers especially. So let's talk a few minutes about cyanobacteria and the toxins that they produce. Let's check my time here. Okay. So we have several categories of toxins. There's way more than this. I know that probably doesn't make you happy, but we're going to talk about the main ones and about the most common toxins. So we have hepatotoxins, that's microcystins, nodularia, and cylindrosperm opsin. These are cyclic peptides, and that has some pretty nasty implications for water treatment and for how you deal with the water once you realize that stuff is there. And neurotoxins, they cause a paralysis. Usually it causes respiratory failure. This usually ends up that you lyse the cells in the intestines and you bleed to death. It's a nasty way to go. Dermatoxins, which look like they might have a little bit more effect than just being rashy on your skin, but that's what a dermatoxin means. Usually happens pretty fast, hours to days. Then BMAA, and this is still, we're still not sure about BMAA and DABA. The interesting thing is that the methods that we use to detect these toxins are not near as well developed for these as they are for these guys, and there's a lot of chatter in the literature about how much of a problem or not a problem BMA is, and it has been anecdotally linked to cells for ALS and Parkinson's and Alzheimer's, as has microcystis actually. Cyanopeptides beyond microcystins and abinopeptins. This is interesting because you can actually get stick tests for this now. They're starting to develop methods for these, but they don't appear, they're not as produced reliably as some of these other toxins are. And so you don't see a lot of reference to them in the literature as being ones that are a problem, but we also don't look for them as much. When you think about it, any toxin can act like a neurotoxin if it's a high enough concentration. So if you have microcystin that is up in the, 60,000 to 100,000 micrograms per liter, which can happen, it's going to act like a neurotoxin. It can be really concentrated. Give you an idea of how toxic these things are. When you look at LD 50/50, a lethal dose for 50% of the population, saxitoxin, not too bad, right? Ricin, cobratoxin, most of the algal toxins are actually more toxic than cobratoxin. Strychnine, curare, actually it takes quite a bit of those. It takes a lot less of algal toxin. So nodularin, something you wouldn't necessarily have on your radar unless you're in a brackish water area. Most of the data is based on ingestion or direct dosing. We don't have a lot of good information about inhalation other than to know, yes, these toxins along with algal cells do get brought into the air by turbulence, and their inhalation is a problem, especially if you have a respiratory issue. So we know that they cause problems. We just don't have a lot of good data about how important that information is. So when you think about measuring toxins, you have a couple choices. You can directly measure the toxins, or you can measure the presence of toxin-producing genes, and that's assuming that you already know that you have a potential toxin producer there, which you really need to know for both of these methods because you'll spin your wheels a lot if you don't. So your choices on analytical methods are mostly ELISA, LCMS, and LCMSMS, and there's also GCMSMS. this is wet chem essentially, and probably the easiest one to do is ELISA, and LCMS and LCMSMS are the most analytical methods. And they're going to give you, if you get a screen for ELISA and you need to have a really good idea, okay, how high really is this, and you're going to move to one of these other methods. The most expensive one is LCMSMS. These pieces of equipment can cost anywhere $50,000 to $300,000. ELISA, you can get started for, by the time you get everything you need, probably $2,000 to $3,000, so much cheaper overall. In my lab, we only do screening tests. We just use strips for antitoxin A and microcystin. So, and we also have cylindrospermopsin strips as well. We made the decision a long time ago to not measure algotoxins. My expertise is biology and taxonomy, not chemistry. I could do it, but it's expensive. And I am busy 100% of the time doing taxonomy. So I'll let somebody who loves to do chemistry do the chemistry, and I'll stick with biology. ELISA kits are available for microcystins, nodularians, saxitoxin, antitoxin A, cylindrospermopsin. So the thing you need to know though is that some of these are cogener specific and non-cogener specific. And right now, I believe that there's only standards for 10 of the microcystin cogeners. I believe it's something like that. But there's like 250 cogeners for microcystin, and that number goes up every single time I do this lecture. So every single time I hear Barry talk about it, that number goes up. So it's complicated, and it's based on, for microcystin and nodularian, on the presence of an attostructure within the molecule, but only about 85% have that. So it's complicated. And anytime you go to analyze any of those by ELISA, you know that you're essentially estimating. LCMSMS, that's using, pretty complicated equipment that, is pretty pricey, on the order of $150,000 to $200,000. You have to have an analytical chemist, a biochemist, run this kind of instrumentation, and it takes a lot to maintain this kind of instrumentation. So I send it out to a lab, and you're essentially looking at something, being held on a column, and then being released based on how hydrophobic or hydrophilic it is, and how much affinity it has for the column, if you're using a column like GCMS, or how heavy it is. So, and you have to make sure that this peak is actually what you think it is, and that's why you always need to have standards. EPA has guidelines. I kind of feel like Pirates of the Caribbean. It's really more of a guideline, which is a little frustrating. I wish they had actual regulatory language when they released this, but they didn't. They're advisories. But what they say is essentially for drinking water, they have microcystin and cylindrospermopsin. They have stratified it by school age, which I understand the biology behind this, but no family, unless you have one child, are you going to be able to stratify by school age. So I pretty much ignore these higher ones, and I always go with the lower ones because I have grandkids now. So I have little guys in my household, and we're not making formula, but my 5-year-old grandson is drinking water out of the tap. And yes, we're filtering that through the fridge, but there's still a possibility that there could be microcystin carrying into the distribution system. So, and then, so you always want to go with, I prefer to go with the more conservative 0.3 micrograms per liter and 0.7 micrograms per liter for cylindrospermopsin. Don't often see high cylindrospermopsin hits. Microcystin is definitely a hit or miss, but about 85% of the time it's producing a toxin. It just depends on whether you can detect or not. For recreational guidelines, this was interesting. They originally had this set at 6 or 4, which was more restricted than Ohio. Ohio is 6 micrograms per liter for microcystin. Ultimately, by the time this came out, by the time they actually released the guideline, it was 8. I don't disagree with that, but I'm not sure I would be swimming in 8 micrograms per liter of microcystin. For cylindrospermopsin, it's higher. It's less toxic, and that's 15 micrograms. They did not stratify that for age group. Realize, though, that little guys are behaviorally at risk. So they, just like dogs, they're in the water, their hands and mouth, they're smaller, so their small body size makes them more susceptible to toxin issues. You need to be careful. If you see a scum, you shouldn't be anywhere near it. You shouldn't be in it, your kids shouldn't be in it, your animals shouldn't be in it. So, the axiom is, when in doubt, stay out. And my when in doubt, it's wider every year. So the other option for us is to actually look for the genes, not necessarily for the algae themselves. If you're trying to figure out risk and you're trying to figure out, okay, do we or don't we have, potential toxin producers there, and you don't have access to a phycologist, then you can look for the toxin producing genes. The problem with that is that you don't always know if those genes are turned on. So, and that's, the epigenetics of what's triggering that particular thing to either turn the gene on or not. Now, they know that there's an association with nitrogen. They know that there is some allelopathic stuff going on, but in terms of being able to truly predict, it's not really possible. And the problem is it's not fast. So they do have really good PCR tests now for primesium, which is the golden blossom, golden bloom that Ken was talking about in the Southwest. And that's because it tends to bloom with unispecifically. It's the only thing there, up to several hundred cells per ml. The problem is with algae is with the cyanobacteria is that they don't always conserve their number of genes in the cell. So it's not one-to-one. And it takes time, and it takes pretty good equipment. And I don't necessarily, I think it's another tool in the toolbox, and I think it can be helpful. It's just that I would rather, if I'm going to assess risk, I would rather have a good count and have a good ID and be able to go from that. To me, genetics informs what you're doing, but we're not at a place yet where it can be applied quickly and evenly throughout the environment. I don't think it's a great thing to dissociate understanding natural history of the organism you're dealing with from the genetics of what's there. So PCR is another genetic technique, and it involves quite a few steps, and you have to have the right amount of material, and you've got to have sequences to be able to actually do the analysis. It takes time. Now, like I said, they've done better with single taxa than they have with consortiums. So when you have something that you only have to look for one thing, and it's a relatively simple, relatively simple community, detecting like primesium actually works pretty well. And they've done the same with microcystis. They're having pretty good luck with microcystis in some cases, but that's usually in places where they know microcystis already is. So we don't know exactly what the environmental triggers are for toxin production. It is staggering the number of new toxins that we keep finding. I can no longer fit my nice little, if I have my full ecology lecture where I talk about all the toxins, they're not all the toxins anymore. They're not even close. I'm now into multiple slides actually. It's probably pretty likely that all cyanobacteria can produce some kind of toxin. Remember, it's just a biochemical, it's just a chemical that we react adversely to, right? So chances are they probably are produce toxins to some extent, or can to some extent, or used to be able to some extent. There's a lot of variability among fish and invertebrates, their impact on fish and invertebrates, and vertebrates as well. There's differences in bioaccumulation. There's differences in deprivation rates. So it might clear a mussel in 10 weeks and clear a snail in 2 weeks, or a fish in 3 weeks, a bass in 5 weeks. We don't have a good handle on that yet. We know that they bioaccumulate. And sometimes they biomagnify depending on if there's other things eating those things, sometimes not. We have a really poor understanding of this, acute versus chronic. So, acute means you drop dead. That's pretty clear reaction, right? Toxins are super high and the organism was super sensitive. We don't have a great handle on long-term effects from low-level exposure. So those anecdotal cells of ALS and Parkinson's and MS, we don't have a good idea of tumor production. We know it takes decades sometimes, but not sure exactly how long. And of course, your physiological, just like with growth and death and growth and loss, your physiological state has a lot to do with how you react to that. So we just don't have a really good handle on chronic exposure and what those effects are going to be like, and the actual degree of impact on humans. If you were in other countries, if you were in China, Australia, South Africa, Brazil, you would be talking about human death. We rarely talk about human death in the US. And unfortunately, it's not because our genetics are any different, right? As organisms, we are the same Homo sapiens sapien, and we react to toxins the same way. The problem is associating exposure with the result, and the epidemiology of it is complicated. Are more algae producing toxins, or are we just now detecting it better? It's probably both, in my opinion. And the ability to produce toxins doesn't necessarily equate to having lots and lots of toxin out there at hazardous levels. So when they did the National Lake Assessment, the NLA, in both 2007 and 2012, they found that there were lots and lots of producers out there. They often were not very high. So lots and lots of systems at or below 1 microgram per liter, right above detection, right at detection, with a few that were super high. So the detector, they're out there and they're producing. They're just not necessarily producing a lot. And what causes them to go over that threshold to hit that tipping point, we don't have a really good handle on. So, and that's Ken's picture, by the way.
All right. Thank you, Anne.
Yeah.
We are a couple minutes over, but we can
Sorry.
spill into the live session if we want to. So we do have one, while we're switching everything over to Ken's presentation, we do have one quick question from Lou Feeney. Do toxin test strips give enough info for lake users, and can you recommend test strips?
I can tell you what I use. I use Abraxas strips. They are a nice screening test. They're not an end all be all. So I'm going to use multiple indicators of high biomass. So I'm gonna use a test strip when there's a fair bit of algae there. They're not great at low levels, even though the tests go 1 to 10, recreational tests go 1 to 10 microgram, drinking water goes 1 to 5. It's still a rough indicator of worry, not worry, contact somebody, don't contact somebody. It's a nice tool for being on a lake association or a quick decision whether to send off a sample, but it is a very rough indicator.
Great, thank you. All right, one more lecture to go before we get into bonus time. So Ken is going to, and I bet a lot of people are waiting for this one. Ken's going to talk about control. So
the
probably
heck
what
with
everybody's
figuring
here
out
for.
Yeah, heck with figuring out what they are, how pretty they are, what toxins they are. I just want to know how to kill them. It's, um, just a comment on the toxicity issue, particularly for those of you working in New England, because I run across this a lot recently and it bothers me. You should be toxin aware, not toxin terrified, and you should treat your clients and colleagues the same way. I've run into too many groups that will tell someone else, whether it be within an association or a consultant working for them or a state agency, whatever, we have found toxic cyanobacteria in your lake. And they say, whoa, 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 back up. You did an analysis of the toxins. Well, no, that was too expensive. Well, how do you know they're toxic? Well, they were cyanobacteria. Well, it's probably true that they could produce toxin, but how do you know it was a toxin of some level of concern? Well, we don't, but this is okay. We're going to post the lake anyway. Better safe than sorry. And it's hard to argue with that logic. However, there is a huge difference between a pea soup green lake in its entirety and that tiny little fringe of blue greens that blows up when the lake turns over in the fall. Very, very different. Now, Ann was right. You stay out of the scum. If you can, when in doubt, stay out. Don't let your pets lap it up because that could kill a dog. But you don't know that it's toxic, and scaring everybody that's toxic is just not good science. I hate to sound like the mayor from Jaws. Oh, it's okay. You can go in the water, but be aware that you could really be hurting property values, people's psyche in terms of what's safe in their lakes. If you're out on Cape Cod, people are terrified to go in the ocean anymore because of sharks. So they're all going to the lakes. And now we've got blue-green algae showing up and they're being told they can't go there either. It's a little bit scary how many people use scare tactics. Again, it seems a societal thing. You can see it in our political arena as well. Be cautious and be accurate in your statements about what's there and whether or not you know it's toxic. Because the reality is, when it's all said and done, even pea soup green with cyanobacteria only has about a 20% chance of having toxins at a level of concern. Now, 1 in 5 is still good enough odds that I don't think I'd want to bet against it, but it's not guaranteed that there's a problem. And we need to be doing the toxin testing to know. So whether you're using strips or a reader or getting the full analysis done, a lysis, whatever, make the effort to find out whether there's really toxin present. I have been consistently surprised at how little toxin has been in some of the bad situations I've seen in the last few years. Okay. We're now going to talk about.
Ken, it's Chris. Real quick, can you make
Yep.
sure we can't see your slides right now?
You
So
can't see them?
no. Do you have them on a different
All
screen?
right. How's that?
That's it. You got it.
Okay. That's the answer.
Thank you.
That's right. Only I need to see them. Okay. The ecological basis for algal control, I'm going to scroll through very quickly because Anna's really covered that. We were both trying to figure out how to make sure we work this in. It really is the same routine. All right. If I can get it to move. All right, the same problem I had when I started last time. I couldn't, there we go. Okay, growth processes, the various ways that algae grow, loss processes, the various way we lose them. Algal blooms or problem algal situations arise when the growth processes overwhelm the loss processes. And everything we do to control them is either trying to reduce growth or increase loss. It really is that simple. Of course, there's a lot of detail to how we go about it. The key factors, you need to know what you've got for phosphorus and nitrogen. You need to know the abundance and size structure of zooplankton because they're the second most important thing in how much algae will be present. Temperature and oxygen depth profiles are extremely useful. pH matters to the types of algae. Dissolved organic matter makes a big difference. Color may be a sufficient measure. And the algae in the upper waters and the metalimnion, just sampling at the surface won't tell us everything we need to know. Okay, there are three major modes, really four modes of bloom formation. One is it washed in from upstream. Okay, we're not controlling that, but that's an option. Organic growth in the upper water column, a little bit is present. There's enough nutrients, it grows into a bloom. Turns out not to be the most common thing. More often than not, it's either forming at a mid-depth if you have stratification and then moving up, or it's forming near the bottom in shallow lakes or at a mid-depth in deep lakes and popping up. So these blooms tend to show up kind of quickly. All right, so now we're into the methods of control. Simple process. Don't lose your head, get your ducks in a row, don't bite off more than you can chew. First and foremost is understanding the watershed and managing it. I get dumped on a lot for making it sound like you shouldn't bother with watershed management. That's not what I'm saying at all. As will become apparent, I'm saying that you're unlikely to solve most of these problems with watershed management alone, but it is job number one. We have natural background levels for phosphorus that are higher than some areas, in some other areas than others. We have land uses that make a huge difference. Anybody know where this is? Of course, you can't respond to me directly. That's actually Mexico City. There's their drinking water reservoir. There's part of the city and a golf course all draining into their drinking water reservoir. Yeah, great idea. But sometimes you don't have a choice. Managing the watershed is not an easy task. It got made to sound easy, or we can do it with just enough effort by a lot of agencies, including the EPA throughout the 80s and the 90s. And I'm here to tell you, it's just not true. Extremely difficult to take a damaged watershed, something that's more than, say, 30% developed for agriculture or urbanization, and make the water quality that comes off of it acceptable. We have source controls to work with, and that's a great way to go if you can, but you're not going to do that in Mexico City or next door to me in Springfield, Massachusetts. Those things already exist. Pollutant trapping, detention, infiltration, uptake and treatment, maintenance of facilities, all these things are important. They're all good to do. We even have regulations that call for them in many areas. The bottom line is once your watershed is developed to a certain point, the odds of you being able to make the water quality acceptable for the lake into which it goes is really reduced. Just the quick rundown, developed land increases phosphorus loading by an order of magnitude, usually greater than tenfold. Common best management practices reduce phosphorus by maybe 50%. There's a few things that'll get better, but not routinely, and you have to apply them all over. Agricultural impacts are at least as great as urban impacts. I'm not here to beat up on farmers. I like to eat fresh produce too. I enjoy meat, sorry. But, the bottom line though is we run a risk when we have the situation we do with our agricultural industry and our urbanization of impacting our water quality. And it is not a simple matter of, oh, we just have to put some VMPs in place. Again, with more than about 20%, yeah, you could stretch it to 30% of that watershed in urban or agricultural uses, the receiving lake is going to be impacted. There's just nothing that works well enough. The bottom line is we cannot, in this lower picture, we cannot make land that looks like the part on the right behave as though it's the land on the left. There is nothing that will do that. I love this. This is actually a study done by Kathy DeBusk, who's got married and changed her name, so I've lost track of her. She's in North Carolina. We had her come to the NALMS conference back in, I think, 05. She's like 15 years old. This is what it took to reach or to attempt to reach water quality standards for phosphorus by reducing the loads through watershed management. This is the cost per square mile, and this is the percent load reduction. They didn't even reach 30%. And they were leveling off after about $3 million per square mile being spent. How many watersheds do you know where they can spend even $3 million per square mile to minimize impacts? Not many. And yet, that's only getting you a 30% reduction. That may not be good enough. So there are very real limitations to what we can do with watershed management. Yes, it's still job one. Yes, you need to understand what's going on in the watershed and how much of the problem in the lake is caused by it. But in the end, you're going to need in-lake management to be able to do anything in the vast majority of cases. I have 2 or 3 projects in my 43 years of consulting where I can say we fixed the lake by fixing the watershed. And I have been involved in thousands. It almost always involves an in-lake solution, which is why organizations like NEA-PMS exist. We know our businesses, it's not like we've created this. This is very real. They need our services. There's a lot of data you need to have something on it to understand the system to properly manage it through in-lake controls. It is not a matter of going out and tossing a chemical in or dredging or whatever else. There's all these things matter. You may not have to know every last detail about them, but you've got to know something about it to have some idea of the predictability of your results. Okay, so the run through of techniques. This goes pretty quick. It'll probably take me at least 2 breaths. In-lake detention. It's basically relocating a lake or a watershed function into the lake. You can do it. This happens to be a small lake in Arlington, Massachusetts, where we took a piece of the lake and created a detention area. And the stormwater went in there and it cleaned it up. We were getting 200 micrograms per liter of phosphorus there and 20 out in the lake. This looks really big compared to the lake. It's not. It's just the perspective of it. Dredging. Dredging is pretty drastic, when you do this in a lake, but I've seen it do wonders for algal mats. And of course, you're removing the nutrient reserves, you're removing the seed bank, which includes algal resting stages. You can get very good control of algal mats by this in most cases. It's very expensive, very difficult to permit in many places. So it's not like this is something that you're going to do routinely. Where it's been done, it's almost never done just for algae control. If you've got enough depth that you don't need to reclaim the depth, it's way too expensive for what you get for it. Harvesting, another issue. The use of a harvester, a typical, back up one, weed macrophyte harvester is not going to work very well for algae. The mats are going to stick to it and roll over and go right back in. If they're really big and bulky, you might, but they do make harvesters with fine mats or even like a tight conveyor belt where you can pick them up. I know the Chicago Botanical Garden uses one. If you're in a situation where you can't control the nutrients, but you want to pick up the mats, you could do that. They run around early in the morning before they open up and make sure the water places are clean. It's a little hokey. It's not really getting at the real problem. It's not something you see done a lot, but yes, it can be done. Dilution. Again, dilution and flushing are interesting things. Dilution is just putting any amount of water in to keep the thing flushing so that the algae don't build up. That's flushing. Dilution is putting in water of a lower nutrient concentration so that you get an average that's lower than you had before. The catch is you're still putting in more nutrients than you would have had because it's a flow times the concentration. So you're loading it up, but you're loading it up in a way that reduces the average concentration. That's pretty tricky, and it doesn't happen very often. Flushing is more often used where you've got a fairly small water body and a big source of water. The example I'm showing you here is actually a reservoir on the Columbia River, and they're diverting the massive flows of the Columbia River through its Moses Lake, and it did work. I've seen it work on a smaller scale as well. But most of our lakes, we don't have that kind of quantity of water to mess with. To make flushing work well, you have to turn the water level over. You've got to completely replace the water in the lake at least every 3 weeks and preferably every 2 weeks. That is not something you're going to see in New England very often. Selective withdrawal. This is an interesting technique, and we've had some pretty good successes with it in certain areas. You're basically pulling the water out of the deep zone of the lake and up and over and out the dam, either by a siphon, a pump, whatever you want to set it up as, so that the water down here is constantly being replaced. If you do it well, this never becomes poor quality water. But again, it means you have to pull the water out at a rate that may cause the lake to be drawn down or may cause it to destratify. And there's a lot of places where that isn't going to work out very well. So again, a run of the river impoundment, this could work. A basic kettle hole sitting somewhere, nah, not going to happen. Dyes, again, dyes get permitted like herbicides, which is just not right. They're not killing anything. They're just decreasing light penetration in the water. They create an interesting color. It's fairly artificial. Looks okay in a golf course pond or out by Rockefeller Center in New York. It's not the kind of thing you do in a recreational lake. I once was involved putting a small pond in for a wealthy family in Western Connecticut, and the woman said, well, the dye in is just spectacular in terms of the appearance. What will happen if I swim in? I said, oh, you'll turn blue. And she was all packed. No, not really. It's again, it's kind of hokey. It's kind of artificial, and it's still going to allow some things to grow. It's going to cause the lake to stratify as shallow as 5 feet. You may get anoxia in relatively shallow water. So it's not perfect by any means, but it's an option. Sonication, this is one that's gotten a lot of bad and good press and mixed reactions. It's pretty clever. You're basically using sound waves to disrupt the cells. We use this sort of stuff in the lab. It's used medically. I mean, it works. Whether or not you can make it work out in the lake at this expansive, range where you need it is up for grabs. Most of the product literature shows you something like the bottom here. It looked like that. We made it look like this. Well, that's not what you're trying to do. You're trying to keep it looking like this and never let it get to that. Some of them, the simpler ones, are just, unidirectional thing that sits there and it shoots the sound waves out. The fanciest ones actually have them pointing in all 4 directions and will have a sensor with it that uses fluorescence to figure out what kinds of algae are there and then sets the wavelength of the sound to be most damaging to that type of algae. That's pretty darn clever and sophisticated, but what happens when it changes a little later? To how fast does this react? If you've got a real mixed bag of algae, are you going to favor some over others? Not all algae are susceptible to this either. So it gets a little bit complicated. I will say I've seen it work pretty well in small controlled circumstances. I've never seen it really do much on a big lake application. Here's an example though from one of my own projects. This is where we didn't have the sonication pointing. This is where we did. It sure did keep that surface clean and did not let the algae build up. Fundamental problem here is, though, if you are successful with one of these things and it keeps the algae down, rooted plants may grow and they're going to block the line of sight and block the sound waves, and now you may get back to where you are. So it may not be a very complete solution, but certainly if you're in the intake area of a water supply or something where you're getting algae accumulating along the rocks and it's unaesthetic, this does have potential. Algicides. Okay, we could go on a long time here. There's many people on this call that know more about algicides than me, so I'm not going to go into this in great detail. Pretty much the 2 basic things we use are copper-based or peroxide-based algicides. There's some use of endofol and flumioxazin and other things, some of which have been pretty darn effective, particularly flumioxazin on filamentous greens, which are hard to kill by any other means. Herbicides being, well, algicides being herbicides can get a bad name in certain circles. You're putting in a chemical, you're killing things. Yeah, we are. But would you rather have a little copper in your system or a whole bunch of algal toxins? I mean, it's not it's an either or situation. Copper gives you rapid results. Dave Hammond was on at the beginning showing some examples of what they've done with Earth-Tech, which is copper-based. You can get pretty darn fast results. And if you are a water supplier who's got to provide this water to people, whether you've got a bloom or not, you need this kind of thing in your back pocket. It'll lyse cells, not always, but often see the contents into the water. You may kill them and get them to settle if it done just right. The formulation affects the time in the solution and the effectiveness. So it's not like all copper is created equal. There's a lot of different formulations. It's really important to think through what you're trying to do, what the goals are, and what the best approach is going to be. It's definitely something where getting professional involvement is worthwhile. It's not a matter of just throwing some copper in the water. Yeah, copper is possibly toxic to other aquatic organisms. At the levels we tend to use it in the Northeast, there's not a whole lot else that's going to be grossly damaged. I've not seen fish kills result. Massachusetts Division of Fisheries and Wildlife did a survey for 20 years and couldn't find a single case where algicides had caused fish kills. In fact, they found very few where any kind of herbicide use caused it. But it is possible, and the worst effects I've seen are actually on zooplankton, because if you let the thing get a little too high, the zooplankton can be impacted, but they'll come back. Long-term buildup in sediments a concern, but there's really been no paper out there that says it's really bad. It could be a problem if you then later go to dredge, because now you've got a high metals concentration. It may make disposal more expensive, but it's has not been proven to be a big problem. Folks in New York have looked at that pretty extensively and reported on it at NEAPMS in the past. We do have some species that are resistant, and blue-greens and green mats are among them, certain ones, so it may not be effective all around. You can put it at the surface. You can also inject it deeper. I find that where we have problems with the blue-greens that grow at the bottom and then pop up, if you treat a donut in the lake with a chilled solution that sits on the bottom, you can often prevent that from happening. We've had it work very well in several reservoirs we've managed. Again, it's less effective at colder temperatures, but it does still work. Peroxide, the other major one, can also lyse cells, tends to be more effective on thin-walled species, less so on diatoms and greens, so you tend to get the blue-greens taken out better. It degrades to non-toxic components, so that part's fairly favorable. Also applied to the surface, but with granular things, you can drop it in and get to a greater depth with slower release. You don't get any unwanted contaminants, but it tends to be more expensive than copper. You got to work on the factors of dose and frequency to see if it works out. I know there's folks in Connecticut in the water supply system that are using peroxide pretty extensively, and they feel they've gotten the cost down to being on par with copper because they're able to do it less often. So there's some playing around to be done. Both copper-based and peroxide-based algicides have merit. There's a catch. They should be used to prevent a bloom, not remove one. If you get called and it's already pea soup, you don't get a lot of choice. But the object is to track when the algae are growing, figure out when the bad stuff is in its exponential increase phase, understand the water chemistry, pick the right form of algicide, and apply it at the right dose in the right places at the right time to keep the bloom from ever forming. I've got a number of places where I've done that, and well, I haven't done it. I've recommended it. Actually, folks at Solitude have done most of these, and it's been very successful. Phosphorus inactivation. This is one that's near and dear to my heart because I've done a lot of it in the last 20 years and been pretty darn happy with the results. Basically, you're putting in a compound that will bind up phosphorus and make it less available. As we've discussed multiple times, nitrogen is important, but nitrogen is more important to the type of algae, not the quantity of algae. It's the phosphorus that tends to control the amount of algae you're going to have. If you can depress the phosphorus, you can depress the amount of algae in total. Inactivation will put in any one of a number of binders. Iron's the most common natural one, followed maybe by calcium in certain higher pH systems. But what we find is aluminum, which is also a common natural binder, works really well because it's not subject to pH very much once it's reacted. It's not subject to oxygen changes at all. And you can inject it and you can use it very flexibly. You can treat inlets, you can treat the water column, you can treat the sediment. Also lanthanum, which is the main ingredient in Phoslock, and that's a really interesting one. Lanthanum is more specific to phosphorus than aluminum. So it is a more efficient phosphorus binder. You may need more of it. It is not a coagulant, so it won't take particulates out of the watershed, out of the water column, and it tends to be more expensive. But there are reasons why this could be a superior product under certain circumstances. The factors in planning the treatments, okay, got to understand the existing load, particularly the internal versus the external. If your lake is a run-of-the-river impoundment where the water is replaced every couple of weeks, it settles down in the summer and sort of festers, but it's based on what got loaded into it a few weeks or a month before. Treating with a phosphorus binder in the lake is only going to buy you a certain amount of time. It is not going to really fix the problem. It becomes a maintenance technique. And I'm not saying there's anything wrong with that. I think in a lot of urban lakes, that's exactly what we should be doing, acknowledging the maintenance need and covering it. But if you understand that you've got an internal load that's really driving the system, kettle hole lakes, things with relatively small watersheds where the phosphorus is accumulated over many years and is now internally recycling, in a one-shot deal, you can buy 20 years of good conditions. So there's a lot of potential here. You have to understand the sources and the activation needs. You need to know how much available sediment phosphorus there is, particularly the iron-bound and the biogenic portion. The biogenic part is just the organic portion that's readily available, and that's a specific extraction test you do. The bathymetry and hydrology of the system works. Is important to understand. Potential water chemistry alteration. You can't let the pH get out of whack. You have to acknowledge metals levels. You have to understand the oxygen concentration. Any potentially sensitive receptors will matter. Fish, zooplankton, macroinvertebrates can all be directly impacted. There can be toxic results, particularly from aluminum, if you don't balance the pH well. We've had plenty of tests where we've not had an issue. There hasn't been a fish kill from an aluminum treatment in like 20 years. We had a few rainbow trout die, a few more than I think should have, in a treatment last spring. And I still can't quite figure it out because nothing else was dying, but these recently stocked rainbow trout did. And we're not sure that they just weren't very well suited to the lake to begin with, but maybe there was an impact. We're not sure. But again, it was 100 or so, or 90 some out of several thousand that were stocked literally the day before treatment. So, there are sensitive receptors though, and you have to be aware of it. Reptiles, amphibians, waterfowl have really not been much impacted, but if they're laying their eggs or something's going on right at the time, people are going to be concerned. So you have to at least take that into consideration. And then the accumulated residues. If you're doing this on a maintenance basis, you will accumulate a certain amount of inactivation sludge. I've never seen it amount to anything I was concerned about. I've heard it sometimes has been an issue at Universal Studios in Florida. I don't know what it is about their water because Disney doesn't seem to have a problem, but you do have to be aware of it. Okay, lake water column treatment. This is one in Connecticut. As you can see, we had outrageous phosphorus levels building up over the summer, clearly coming from the sediment and mostly from what seemed to be organic decomposition, but it also had a really nasty watershed with a lot of stuff in it. So we started doing these low dose treatments. These are milligrams per liter over this lake. They're not big doses. And boy, it knocked it down, knocked it down, knocked it down. And we're down to almost where we want to be with 2 small doses a year. It's maintenance. If we stop, it'll probably go back to what it was within a year or two. It's not as satisfying as managing it in the watershed, but it works. And we got no prayer of making this watershed do what we want. We can't get cooperation from the state or the town. So this is a case where managing it in the lake has real potential. Tributary injections is one I've been doing for over a decade now in Wellesley. It actually doesn't look like this. This is one of our early tests, but you can't even see it going in now. It's so minor. But basically, we have 2 lines that go out to 2 parts of the 2 different major inlets to the lake, and we dose them just when it rains, just from about Memorial Day to 4th of July. So we clean up all the water getting into the bottom part of the lake by then, and we've had fantastic clarity most of the time. Here you see this is before treatment. Clarity was hardly ever 10 feet or 3 meters. Phosphorus was usually above 20 micrograms per liter, which is a problem here. When we first set up the system, it worked really well for 3 years, but we were using alum adaluminate, and it was eating up the equipment. We had a whole bunch of other issues, some of which were human error, and we didn't really do so well for the next 3 years. So we revamped the system. We used polyaluminum chloride, which is really easy to work with. We automated the system so nobody has to go out and turn it on. It comes on its own. I can track it on a cell phone, and you can see the results since 2014. They're just phenomenal. The place is good. The last couple of years haven't been as good because we've been experimenting with how little can we use. And it's still, we're getting through the swim season. Everybody's very happy with the results there, just treating the inlets. They're working in the watershed, but the town only controls 22% of it. So there's limits on what they can do. And then finally, lake sediment treatment, which probably more people here know about than the other things, where you're basically just taking a long-term buildup in the sediment, dosing it in the water column, letting it settle and inactivate that sediment. This is pretty much a tried and proven technology. We do not know everything we need to know. Picking the dose is still a little sketchy. You'd like to go higher than lower, but it costs more. And if you go too high, it interferes with itself and you may get less of a result. The current thinking is if you're more than about a 50 gram per square meter dose, think about splitting it into 2 doses and spread them out over a couple years. Seem to get better results for that. And we're experimenting some with that now. So there's still more to learn. It is not a perfect solution, but where you've had phosphorus buildup from the watershed over many, many years, and the current inputs are really not enough to make a difference in any given year, this sort of thing gives you back excellent conditions quickly and for a fairly long duration. This is Hamlin Pond, kind of the poster child for it. Really poor Secchi disc in the summer for a number of years before the treatment. It had a duck farm on it for 30 years. That was long gone, but the lake was still a mess. Hydrogen sulfide in the bottom, horrible cyanobacteria blooms. Town put a $7 million beach on it and couldn't even use it. They did this treatment in 1995, and the Secchi disc just shot up. You can see it kind of declining over time, and it like all went bad all of a sudden. 2014, well, the very end of 13, there was a bloom in like late September, early October, and then 2014 was bad again. So they treated again in 2015. You can see we're doing great again. So this has worked well, and we got 18 years out of it there. 20 years is about the average you get for a deep lake. Tend to get more like 10 years for a shallow lake. But again, when you divide the cost by what you're getting for it and the resulting conditions, it tends to be worth it. The aluminum longevity is a function of external loading still coming in, release from phosphorus and organic matter that may not have been inactivated when you did the treatment, and upward migration of sediment iron-bound phosphorus through the zone that you did inactivate. So there's a bunch of things going on at once here. The upward migration is the one that kind of gets you in the end. That's why you get maybe 20 years out of it, because you can't do much better before that gets up there. The other two can make it so that the treatment results aren't as good as you'd hoped or don't last as long as you'd like. So understanding these is pretty important. Okay, lanthanum modified bentonite clay, Phoslock. I don't have a lot of personal experience with this, but I know a lot of people who do, and I like the product overall. It's developed in Australia. It's used more in Europe, some in Canada, a little bit in the US. I know there have been treatments done within the NEAPMS zone. There's no direct pH change. You don't have to buffer it. It's pretty specific to binding free phosphorus. It's pretty stable once it's formed. It doesn't seem to have any toxicity issues. CPRO is the one that markets it on behalf of Phoslock in the US. I like the idea of the clay coating. It's put in with bentonite clay. If you can get enough of that on top of the organic sediment, you can reduce the oxygen demand. Now, I don't know if that really happens or not. It hasn't been well studied, but the potential is there. So it's another way to go about it. Keep in mind, if you have a problem in the water column with actual algae, the aluminum will bind them up and help settle them out. Phoslock isn't a coagulant. It will take out free phosphorus in the water column. It won't take out phosphorus already bound up by algae or other particles. And then it's going to attack free phosphorus down in the sediment as it's released. So the Phoslock might give you a longer term result if you have biogenic phosphorus being released year after year after year. There's still more work needed. It does not have as long a track record as aluminum, but overall it seems to be pretty positive. Lake Lorraine in Washington was an example that was given to me by the CPRO folks. They went from the top picture to the bottom picture. I have heard that it has since gone back. I don't know if it's been retreated. I don't know why that was. Again, none of these are permanent solutions, but look at the watershed. You're not going to make those people go away. You're not going to get the nutrients to disappear. So you have to do something about it in the lake. Okay, selective nutrient addition is one that admittedly scares the hell out of me. There's great literature out there showing that if you manipulate nutrients, going back all the way to 1973, messing with silica, you can prolong diatoms. Increasing the nitrogen, you can shift things to green algae and away from blue greens, but you're still going to have a lot of algae. They use this in fertilization for fish production, and they're not trying to cause blooms. They're just trying to bump up overall productivity so there's more algae, more zooplankton, more fish. Again, there's nothing wrong with high productivity as long as something's eating it at the next level up in each stage. The problem is when you get something that's inedible, like a lot of our big blue greens, and they just take over the system, now you have a very energetically inefficient system and a whole lot of other problems to go with it that may be harmful to the fish. So the biological structure is really important when you're playing around with nutrient ratios. I personally do not have the guts to go tell an association, oh, just add some more of this, it'll get rid of your problem. Pretty much we try to reduce nutrients, not increase them. But it does tell us that there is potential for this, maybe in places where you can't possibly control the amount of phosphorus, a golf course pond, something sitting in the middle of a whole bunch of septic systems. Maybe there are places where playing around with this makes the most sense. Okay, oxidation and circulation could be the subject of a very long lecture by itself. There are many different means of getting oxygen into the water, circulation being one of them. There's a whole lot going on here, and just to make a claim of oxygenation or circulation is not adequate by itself. You have to understand the details of the process. So, oxygenation and circulation, again, all circulation tends to be oxygenation. Not all oxygenation is circulation, but it can work by adding oxygen and facilitating phosphorus binding. It can alter the pH and the related water chemistry in a way that favors preferable algal forms. It can create a suitable zooplankton refuge that will enhance grazing. It can also destroy them, as some recent papers have shown. And then circulation by itself will create turbulence that may neutralize the advantages conveyed by certain buoyancy mechanisms, particularly in the cyanobacteria, which may shift you away from cyanobacteria and into something else. Just be aware that not all cyanobacteria depend upon buoyancy, and you may get other cyanobacteria in place of the ones that you knocked out. And finally, homogenization yields consistent water quality, which may not be what you're looking for in a recreational lake, but water suppliers will take consistency over a slightly better water quality that's more variable every time because they can treat it and deal with it. Okay, non-destratifying oxygenation comes in 4 major ways. The simplest is basically dissolved oxygen injection. If you have a deep enough lake that those oxygen bubbles can be absorbed before they get to the thermocline, so they will not cause destratification, this can work. It's the simplest, easiest way to do it. It's one of the oldest ways, but it was not until we developed better delivery mechanisms and had more oxygen produced and had on-site oxygen production that this became economical and beneficial on a longer-term basis. But it doesn't work if you've got a lake that only has 5 or 10 foot of hypolimnion at the bottom because you don't have enough runtime to get all the oxygen dissolved. So then you need to either bring the water into a chamber and oxygenate it or aerate it, or pull it out of the lake and oxygenate it or aerate it out on shore and put it back. Basically, these hypolimitic aeration chambers were used for many years. You're seeing them less and less frequently now because they're energy inefficient, but they work. You're putting air, you could do it with oxygen, into a system. You're pulling the water in, it's oxygenating over a vertical rise, and then the water goes back to where it came down the outside of the chamber. It's effective. It isn't efficient. If this breaks down and things go anoxic, then you got problems. And of course, your equipment's in the water, which makes it a little harder to maintain. Same thing for downdraft bubble contactors or species cones. They're under pressure because they're deep already, so you get tremendous transfer. You're putting oxygen bubbles in that want to go up. You're pumping water in that wants to go down, and you want to balance it so that all the oxygen gets absorbed. Otherwise, it's got to go up and be wasted to the surface. But these have had some super success stories. The problem is you need this major structure on the bottom. It's got to have a platform that it can sit on so it doesn't settle into the sediment. If you maintain it, you've got to go down there with divers. It's expensive, it's efficient, and it may be effective, but it hasn't been favored in many cases. You can, however, pull the whole thing out of the lake, put all your equipment on shore, pressurize the container like you'd get naturally if it was sitting in deep water, add oxygen to it, and then put the water back in. That is really effective and can work in very shallow hypolimnetic situations, but it's more expensive. Also, if you don't pressurize and oxygenate quickly and get it back, you're going to get an increase in temperature, and now that water is going to want to rise. And there have been some disasters created by that. But this is the side stream supersaturation is a technique that's coming into its own. You're starting to see commercial versions marketed. If you don't have a deep enough lake to let dissolved oxygen work by itself, this may be the next most effective way to do it, and it's easy to control. Side stream supersaturation is one term. It wasn't sexy enough for one group, so we came up with nanobubbles, which you're hearing a lot about lately. It's sort of a version of this. The difference is here it's pressurized, and you're pretty much guaranteed that those oxygen is actually dissolved in the water. Nanobubbles are bubbles in the water that are so tiny that, technically they're so tiny they qualify as dissolved by the definition of dissolved. That gets to be a real sticky issue. But the claim is they're really bubbles. They're not dissolved. And they're so small that the surface charges on them hold them in solution. They do not rise and disappear. They're as likely to go laterally as they are to go vertically. That's all believable. And then you've got to get enough of them into solution to put out the oxygen capacity you need to put them back in the lake. In the cases to date, and there aren't that many out there yet, we're still learning, where they've not had to worry about it being stratified, they're just trying to get more oxygen into the system overall, it's worked fairly well. Where we've had stratification and the object is just to oxygenate the bottom water, we've had less desirable results. The primary claim has been that they're not actually getting the transfer of oxygen into this chamber that they want. And what we're putting out here really isn't as much as they think they're putting out, but what gets out there seems to help. So I've got one on the Cape that we've done now for 2 years. It has not worked out particularly well, but a lot of it's failure of implementation, not failure of the theory behind this. It's just a technique that's not quite ready for prime time yet, and we're going to need to mess with it a whole bunch more to get it there. Okay, the biggest challenge to successful oxygenation is induced oxygen demand created by water movement. If you push water across the sediment with oxygen in it, whether it's being moved by the air or being moved by a water current, you get the oxygen where it's needed, but you also cause more oxygen to be demanded. The best way to think about this is if I put a whole bunch of ping pong balls in a pool, kiddie pool, and I put you in there with a net and say, go collect as many as you can in a minute, you'll get a bunch. But if I was able to stir the pool and have it all moving so they all came to you, you could get a lot more in that minute. So that's what's happening here. So whatever you think you need, when you put the oxygen in, you tend to need more. You basically create an induced oxygen demand. So for oxygen, pure oxygen systems, we usually think you need about twice as much as what you might calculate. For air systems, you might need as much as 5 times as much because it's only 1/5 oxygen. Key factors in oxygenation, you got to get enough oxygen to counter the demand and that induced oxygen demand along with it. You've got to maintain the levels suitable for the target aquatic fauna or high enough that it doesn't let the chemical reactions occur that you're trying to avoid. And that number is about 2 milligrams per liter. For fish, you want something more like 5 milligrams per liter. You've got to have enough phosphorus binder present naturally, iron, calcium, or aluminum, whether you're putting it in or it's naturally there for it to bind up the phosphorus. And you don't want to break stratification if that's part of the goal. Destratifying oxygenation or artificial circulation is most often just done with diffused air. And there's all kinds of ways you can do it. There's a zillion things out there. There's lots of companies, including a number of them that sponsor and exhibit with NAAPMS, that know what they're doing with this. It's tried and proven methodology. There's still a certain amount of trial and error that goes with it. There's still some playing around to be done. There's more to be known, but this is stuff we know how to do. If you don't mind having the whole system mixed, we can make this work fairly well. If there's not enough phosphorus binder, there's still an issue. If we're actually causing poor quality water to be mixed into the upper water column, well, there may be another issue. It often doesn't result in lower oxygen, or excuse me, in lower algae than we would like, but we usually do get a shift away from the blue-greens. And if your biological structure is good, then they can eat the other algae. But then you've got to worry about the biological structure part. It doesn't have to be done with diffused air. There are downdraft systems. This is one by Resmix, largely an Australian company, but there's a few in the US and a bunch in other countries where they pull the water down. You're taking oxygenated water from the top and pushing it to the bottom. That's philosophically pretty desirable. The alternative is an updraft system, Solar Bee being an example, where you're pulling the water from the bottom to the top. That's okay as long as you pull enough water that you keep the water circulated well. If you don't circulate it well enough and the bottom water becomes of poor quality, now you're pulling it up to the top where it can do some real damage. Solar bees claim this long distance circulation where they pull the water up, it goes laterally across the top and then eventually goes back down. Well, that eventually is pretty darn quick. It doesn't go that far. And you tend to get a donut around it. And it's usually a lot smaller than what was being claimed in the literature. So there's an issue there where, yes, you get pumping, but these are usually not put in at a density high enough to actually get the job done. And that's a constant source of aggravation going back and forth between the company and practitioners and so on. But the theory's sound. It's just that it doesn't do everything they say at the level they claim. I want to mention their fountains also are the same idea. You're throwing the water in the air, you're oxygenating, putting it back down, aesthetically pleasing, not very efficient, but it could be effective. Okay, the biggest challenge in successful circulation is maintaining the mixing. Water warms through the summer. You're not getting rid of heat from this, you're just mixing the water. So the water's getting warmer and warmer. It's a simple physical fact that more energy is needed to mix warmer water than colder water. For every temperature degree difference, when the water's warmer, you need more energy to mix it. So this gets harder and harder and harder to do as the summer goes along. Almost no one installs a circulation system with enough power to mix come August or early September. It just doesn't happen. So you just have to hope you get through the summer or it's not too bad too fast, because otherwise you've got to build an oversized system that you don't need most of the time. And that's generally not what happens, but it can work. And the key factors, you got to move enough water to prevent the thermal gradients from dead setting up. That means no more than about a 3 centigrade degree difference from top to bottom. And that's the easiest way to measure if you're successful. General guide, if you're using air, 1.3 cubic feet per minute per acre served. You might need more than that. 2 cubic feet per minute per acre is a good target. If you could, that's a little more oversized. If you're doing it by pumping, you need to move 20% of the target volume per day. So about every 5 days, you've moved it all. Could need as much as 100% per day. It's a question of how warm it is and what the conditions are. Almost none of this is going to overcome high nutrients if you've got no control of the nutrients, though. You may get different types of algae, you'll still get a bunch of algae. You have to balance the delivery of the oxygen near the bottom of the lake with avoiding sediment resuspension, and that's a difficult challenge with these systems. And then you've got to move the surface water to a depth that's 3 times the Secchi depth if you want to actually lower biomass so that they're spending more time in true darkness. In a shallow lake system, you really don't have much of a way to do that. So you really have to expect you're going to shift the types of algae, and then it's a matter of biological structure, whether or not they can get eaten and whether or not you have a bloom or everything works out. Okay, just a few other ones. Barley straw, I mean, it's basically an unlicensed or unregistered herbicide, but it can work. The barley straw decays into material that or compounds that seem to have allelopathic capability, and particularly are damaging to blue-green algae, so there's some benefit to that. The guy in the pictures here is Steve McComas, who said it best. He said, it works sometimes, but I'm not really sure why. It's not as easily controlled as a lot of other algicides, and frankly, a licensed herbicide applicator can't use it because it isn't a licensed project or product. Viral controls are really popular twice, once in the 70s, once in the early 2000s, at least on the research front. We've never really developed any commercial products that work. And I think the real reason comes down to this. You have your untreated, is just horribly green or dark. Your treated looks like that. Are you really going to be any happier with your lake looking like that? You know, it's hard to get complete control with biological means. Okay, bacterial additives, again, could be the subject of a whole course, which I'm not really qualified to teach. There's so much to be known here, and a lot of it isn't known because there's virtually no peer-reviewed papers on how these things work. Most of it's proprietary. You get a simplistic claim that they allow bacteria to outcompete algae. That's not really that likely. When you get shown an image of here's what it was before and here's what it is 5 or 7 days later and it's gone clear, there are no bacteria that can outcompete algae in 5 to 7 days and then disappear. It doesn't happen that way. You can get a potential organic sediment reduction. They often pair it with circulation or oxygenation because it does work better when the oxygen's high. We get variable results. We have inadequate scientific documentation, but I will tell you there's successes out there. I've seen it work very well in many places. I myself used muck away. I didn't get less muck, but I got way less dense muck. It definitely ate up the organic component. It's a potentially effective, but not necessarily reliable approach. And by reliable, I mean that in the engineering sense, that can you guarantee that you're going to get what you expect? And the answer is no, because usually you're putting in 1 or 2 products. You're just buying something that's supposed to work, getting it applied, and there's not a whole lot of monitoring that goes with it. There's not a whole lot of testing. Most of this stuff was developed in the wastewater and the sludge handling industry. And they're doing a lot of testing and a lot of manipulating, a lot of tweaking to get the best results. We just throw the product in and expect it to help us. That's really not that effective a way to go. The best explanation I've heard of this is from Pat Sims-Geiger's Diversified Waterscapes. He's been to NEAPMS before. He's from California. 4-step process. Use an algicide to kill the algae. Secondly, use enzymes to break down the long chain hydrocarbons, their cell walls and such. Allow bacteria to metabolize the shorter chain hydrocarbons. Helps if you have oxygen. And then use a settling agent to drop out the particulates. That's what clears the water and keeps it clear. If you're using one product, you can only be doing part of this. I believe that a lot of the enzymes that are put in actually work like algicides. Now, it makes them unlicensed or unregistered algicides, and that creates a lot of heartburn in the industry because nobody wants to have to go through that process, and I get that. But the bottom line is you've got to look at it as this 4-step process. And if you're only using one product, you're doing part of the process. Maybe you'll get results, maybe you won't. It's a little harder to figure. Okay, biomanipulation, near and dear to my heart. I made money doing my PhD work. My PhD research wasn't on biomanipulation, but I did it, and that paid for my PhD research. Basically, if you've got lots of big zooplankton, they're the cows of open water. They chow down on the algae, and they can filter it pretty clear, as long as the algae aren't things they can't eat. Smaller zooplankton, like this Bosmina or these Chidurus, eat a lot more. The power of filtration is related to the cube of body length. So being bigger makes a difference. Size matters in this. However, if the phosphorus level is too high, 80 parts per billion or micrograms per liter is usually thrown out, that's going to cause problems. You just can't overcome that much fertility. So you have to keep the phosphorus down to a reasonable level, but then the biological structure makes quite a bit of difference. Balancing the game fish with the panfish, with the zooplankton, with the algae, so that you're getting good productivity and not a bottleneck at any step is what you're going for. So that means now you need fishery scientists. You got to really look at your lake in terms of its fish community. This is the brand newest stuff I've got in here, and I'm really happy with this. These are lakes that have been treated with aluminum. They do not have phosphorus problems. These are like within the first 2 years after treatment. But if there's alewife present, they are eating all the zooplankton. I mean all of it. There's just like nothing there. The biomasses are really tiny. And as a result, even though the Secchi dip isn't awful, at least 2 meters, it's not nearly as high as when we have lots of bigger Daphnia out there. And does happen in the lakes where the alewife are absent. Same statement would go for lakes with gizzard shad in them and a few other things. Golden shiners can be particularly damaging to the zooplankton. If your planktivorous fish community is really dominant, you're going to have less zooplankton and you're going to have more algae per unit of fertility as a result. So if you can keep the fertility down and you can manage your fish community to get more big Daphnia, you will be doing way better. Rough fish, same deal. They stir up the bottom. Some of that becomes usable nutrients. They make turbidity, if nothing else. Controlling them if they get out of hand can be a benefit. And then finally, rooted plant assembly. If it's really dense with rooted plants, you usually don't have algae blooms. There you got another problem. You're going to get called in anyway. So getting the balance is right. Again, it's really more about nutrients. The biology is the icing on the cake. It's not the cake itself. And there's a few techniques that really didn't quite make the list. You can't do circulation with the minivan. Stale bread is not the same as barley straw. And for gosh sakes, never mix Viagra with algicides. Okay. So the roll call for algae control, watershed management's always job one, but it's not always going to get the job done by itself. In fact, it's rarely going to get the job done if you already have a problem. Phosphorus inactivation for internal load control or managing inputs can be really helpful. Circulation to 3 times the Secchi depth, if that's possible, is useful. Circulation, possibly with inactivators or dyes or bacterial additives, can do well in a shallower lake. Oxygenation, mainly for deeper lakes where internal loads are dominant. Dredging where it's feasible, especially for algal mats, but the expense and the permitting are just so difficult. Algicides with proper timing and limited uses. I would say that if you're treating more than once a year, it's too much. You really got to look for a way to control the nutrients. But if every so often you have a problem and you deal with algicides, you are not a bad person. This works. This is effective, and done right, it can be effective for most of the year. I've got lakes where we hit them in June and we're good for the year. We've stopped the problem. We didn't wait for it to get green. We hit it when it was starting. Sonication for susceptible algae, particularly where nutrient control is limited. Golf course ponds, manure pond, farm ponds, things like that can be pretty effective. Biomanipulation, as long as the phosphorus is not too high and you don't mind some wide variability, you can get a lot for it. And of course, if your fishing's an interest, it all works together. And then any other technique as the scale and circumstance dictate. I don't like to see techniques bad-mouthed or thrown out. Everything has its place in the toolbox. Just don't put in screws with a hammer and don't try to put in nails with a wrench. Pick the right tool for the right job. And that is it.